Okay, great. Sorry about that, and I'm back. Um, Coming back to the sessions and what we cover in that. So I spoke about GCP, good clinical practices. I see GCP. We were going to cover ethics. In ethics, like I said, we cover the history of clinical research. We talk about vulnerable populations and special populations, and what needs to be looked at when we are considering them as participants in clinical trials. Then we talk about PKPD, pharmacokinetics and pharmacodynamics. So when I was still talking about pharmacodynamics, as we said, how does a drug act? What's the mode of action of the drug? That is basically, if you don't understand the pharmacodynamics aspects of it, how the body, what the body does to the drug is the pharmacokinetics part of it. And what the drug is doing to the body is basically the pharmacodynamics part of it. Can I request everyone to go on mute, please? Um, Thank you. So, um, so these are various aspects that we essentially covered during this session. And then after that, I think you will be having an internship um, for, I guess, 2 or 3 months, Gayatri, where you will actually work on some of the applications related to drug safety. So you would get some hands-on experience. And that would be the whole course. In addition, of course, you have assessments which go along the way. So what's at the end of this, actually, what's important is what opportunities will you have? And I heard some of you say that you were really looking for different opportunities. And when you're talking about opportunities, there are diverse things that you can really do once you have a stronger understanding of clinical research. And as I can see, some of you already come in with strong backgrounds, either in pharmacy or in clinical research, which is great. In terms of opportunities, there are different areas you can get into. There's, of course, drug safety itself. You can get into different areas as well, such as medical information management. You could go into medical review. You could go into areas such as um, beyond drug safety. You can also get into core clinical research. What this program is really doing is setting the stage for your entry into a clinical research program. And if you look at the way industries are hiring over here, I think drug safety is, of course, one of the top areas. But the ability to move into different roles, to move into the role of a clinical research coordinator, because you've understood this, we're going to look at different roles and functions. We're going to talk about what happens at the site, what does an investigator need to do, what does pharma or the CRO part of it involve, and different roles and functions. So the other options are, if you could go into being a clinical research coordinator, you could get into medical writing, where you're involved in authoring clinical regulatory documents, or you could get into publications. This is not an instant recipe to get into all of these, but this helps set the stage to make an entry into any of these areas. And then depending on what background you have to complement this, it would really work out well. Depending on which area, which geography you're present in, and I assume that most of you would be in the US, but depending on which area you are, there is a lot of, especially if you look at the East Coast, for example, areas like Boston, like New Jersey, or West Coast, there are so many opportunities. You could even enter areas such as clinical data management. I think the opportunities are diverse. You have to find out what is your calling. What do you like to do? Do you prefer to have more of an in-house authoring, analysis kind of role? That's where perhaps medical writing or drug safety could be more interesting, or medical information management. Or do you like going out to the site? And I think, Pooja, you mentioned you were a CRA in the past. So that's when, if you're like traveling, you're going out, interacting, engaging with people. I think moving into monitoring or a CRA role becomes more attractive. So really map your persona and what the industry needs, what appeals to you, to make it a win-win. So would any of you have any questions right now? Okay, so if you don't have any questions, what I'd like to do is to really start with our session right away. And when we're starting with our session, the first topic I'm going to pick up is basically the historical perspective of human research. And so before we start discussing the different aspects of the ethics of clinical research, I would like to ask you a question. Why are we basically conducting clinical trials? And what are clinical trials? Questions. Would anyone like to share their views on that? Feel free to unmute yourselves or to post your responses on chat. What are clinical trials? Any thoughts? To prove that, yes, Sylvia. So to prove that a drug is safe and effective in patients. So, okay. So when you're saying you're going to prove that a drug is safe and effective in patients, so with that, I think the first thing you're hearing is that this is research that is establishing the efficacy and the safety of the drug in humans. So we have crossed the preclinical stage and we are moving to the clinical stage. So efficacy and safety in humans. So that's one aspect. Is it always in patients? Any thoughts? No. Okay, Pooja. So then when do you have it in something other than patients? If you're talking of humans,
Yeah, in BA studies, we have healthy volunteers.
right? Healthy volunteers. Thank you, Pooja. So basically, how many phases are there in a clinical trial?
4.
4 phases, very good. So phase one, two, three, four. So you've finished your preclinical, you've finished your animal studies, and now you're moving to humans. So uh, at each stage, so between preclinical and clinical, you need an approval from the FDA to conduct trials in humans, to conduct clinical trials. For obtaining that approval, you need to submit an application to the FDA, and that's called an IND.
Application.
I'm sorry?
Yeah, investigational new drug. The new drug application.
Very nice. Investigational new drug application. Remember, an IND is your permission to conduct clinical trials in humans. Then you have 3 phases that you go through: phase one, phase two, phase three. So phase one is basically the first time. I mean, there's also phase zero or microdosing, and sometimes it's phase five. At this stage of the program, I'm not going to go into all of that. We'll discuss that in more details later. But for now, we can just look at phase one, two, three, and four. So phase one is basically let's consider that's the first time you're testing the drug in humans. So what would you want to establish first, safety or efficacy?
Safety.
That's right. Very good. So you want to establish the safety of the drug. So why do you think you want to test the drug in healthy volunteers if you want to establish safety? Why not in patients? And I can see that there are a lot of participants on this call. Do request everyone to share their views and participate. Yeah. Why are we focusing on safety? Right, but would you not be able to establish the safe dose in patients? Why healthy volunteers?
What about see if they experience any side effects?
That's correct. So that's what you're trying to establish. You're trying to see that does this drug cause any harmful side effects, any toxic side effects, right? Any adverse events or adverse drug reactions. But why can't we test that in normal patients? Why are we looking at healthy volunteers? You're putting someone who's a nice, healthy human being at risk by testing the drug in healthy volunteers. Why are we doing that rather than directly testing the drug in patients? Now, let's look at what's the difference between a healthy volunteer and a patient. A patient already has complications, has various comorbidities, right? And when you want to test the safety of the drug, establish a baseline for dosages for the drug, that is correct. But my question still remains, why healthy volunteers at phase 1? So phase 1 trials are also called human pharmacology trials. And what you're trying to establish is the safety. And how are you trying to establish the safety? Why healthy volunteers? Basically, when you're looking at a patient, patients normally would have various comorbidities. By comorbidities, I mean they could have associated complications. And when you're looking at multiple complications that exist, uh, hold on, when you're looking at various comorbidities, it can make it far more complicated for you to determine whether those complications are a result or associated with the with your drug, or those are related to other conditions that are existing already and for developing further. So you want to kind of baseline it, as you said, at Indo, in a way that you want to um, remove other complications. You want to have a healthy volunteer who's not having 10 other complications and see if you can determine whether your drug is having any side effects, right? So that's the primary reason why you're looking at healthy volunteers, though there are certain exceptions such as oncology trials, etc., where you may actually test the drug directly in patients. Okay. Okay. So we're seeing that in phase 1 trials, you're testing it in patients. Normally, you would have already established, excuse me, geez, establish a certain safe dose based on your trials in animals. So you normally establish what's called NOEL, no observed adverse effect level, and we'll discuss NOEL and MABEL and stuff like that later. But you've established some kind of dose which you think is safe in animals. Now, will you directly use that dose in humans? Yes or no? What do you think? If you've identified a certain dose that you think is safe in animals based on your animal trials, your preclinical trials, would you directly use that dose in humans?
I would think you'd use a higher dose.
A higher dose. Why do you think you'd use a higher dose?
Because humans are larger than animals.
Because of the weight of the
That's
does it comes with weight. So the humans are bigger than the animals. Than this.
very good responses. So what you do calculate is something called the human equivalent dose, or HED, based on body surface area. So you proportionalize out your dosing based on the area to account for that. But you're still, while you're looking at, you're proportioning it out based on body surface area, but you're not increasing the dose per se. So what you actually do is you apply a safety factor, which results in, at the minimum, a 10-fold reduction in what you had calculated as your human equivalent dose. Because you want to protect humans, so you're starting with, actually, sometimes people go down to something like up to 500 times lower, but usually the minimum at least is a 10-fold reduction in what was considered to be the acceptable dose in animals. So anyway, parking that for a moment, going back to phase 1 trials. In phase 1 trials, you're now trying to establish a couple of things. You're studying, as I think, Pooja, you had mentioned earlier, you're studying the pharmacodynamics, the pharmacokinetics of the drug, basically seeing how it's being metabolized, how is it acting in humans. Remember, even when you're establishing your dose in humans, what is important to identify when you're comparing what you determined as the dose in animals to versus the dose in humans, you try to identify what was considered to be the most pharmacologically relevant species, and then looked at what was the acceptable dose in that species. So, and then you try to identify your human equivalent dose and fine tune further. But all said and done, humans are humans and animals are animals. So the way humans may metabolize the drug, or the way the drug may act on humans, may vary. And that's why it's so important to repeat PK, PD, pharmacokinetics, pharmacodynamics, and try to assess different aspects of this in humans. And you do that, and you also try to identify what is considered to be the acceptable dose in humans as well. So that's your phase 1 trial, and your primary focus is purely on safety. Phase 1 trials are usually unicentric, a single site. Now, what's a site in a clinical trial? Any thoughts? Oh, good, a doctor's office. So it's basically, a site is a hospital. It could be a clinic or a hospital, in simple terms, as you put it into a doctor's office. Yeah, but basically, site is considered to be the institution or the hospital where the trial will be carried out. So terms that you need to get familiar with are: A is the site, B is the investigator. Who is the investigator? No, the investigator is the doctor. Investigator is the doctor. So you have something called a PI, which is a principal investigator. So that's the main guy, or the main person at the hospital, who's taking full ownership for the conduct of the clinical trial. Who is a sponsor in a clinical trial? So, right, Dindo. A sponsor is the drug company. The pharmaceutical company is the sponsor. So the Pfizer, a Merck, a Roche, or an Amgen are all examples of sponsor companies. That's the company is actually going to discover and develop the drug and then market it. When the company has discovered the drug and wants to actually carry out the clinical trials, the drug development process, many times the sponsor company will outsource its activities to a CRO. What's a CRO? A CRO is basically a contract research organization. A contract research organization, you can call it clinical research organization as well, but it's primarily referred to as a contract research organization. The concept is the same. Part or all of the activities related to the conduct of the clinical trial can be outsourced to the CRO. Examples of CROs, there are so many big CROs. Anyone wants to name some of the CROs? You have Syneos, you have IQVIA, you have Parexel, correct? That's IQVIA, Parexel, Covance, Icon, PD. Request everyone to stay on mute, please. You're welcome to unmute yourself to respond, but if you're not responding, then kindly stay on mute because this session is being recorded. So yeah, so these are some examples of CROs. And the work that is performed by the sponsor is outsourced to the CRO. The CRO will then conduct the clinical trial on the behalf of the sponsor, and the sponsor was the pharma company. What does the CRO or the sponsor do where whoever is, whichever of them are actually conducting the clinical trial? One of the first things they will do is identify sites. And what was the site again? The site, correct. They were the hospitals where the clinical trials will be conducted. They will go and identify a whole bunch of sites. So why do you need to identify multiple sites? Because you need to, what does the site do? What is the most important resource that a site provides to the pharma company? Not even the human samples, it's actually the humans or the subjects. So in a clinical trial, ideally you refer to the participants as subjects or participants and not as patients. Why would you do that? Because as we just discussed, it could include either healthy volunteers, or it could include patients. So you refer to them usually as subjects or participants. Exactly, Pooja. Healthy volunteers or patients. And so you're referring to them as subjects. So now you understand who's an investigator. Now at a site, you have the main doctor who's responsible for the conduct of the trial at the site. That person is called the principal investigator. Apart from that, the principal investigator may be supported by sub-investigators who support the principal investigator in the conduct of the clinical trial. You may also have something called a CRC, which is your clinical research coordinator, correct? So sub-investigators and study coordinators. So the CRC is the person who would perform a lot of the activities of the clinical trial, and the principal investigator may delegate some of the activities of the clinical trial to the CRC, to other sub-investigators, etc. Another important piece in this story is the IRB. So what's an IRB? An IRB is the Institutional Review Board, also sometimes referred to as the IEC or the Institutional Ethics, correct, the Institutional Ethics Committee. So this is a body that's usually associated with a site, which is the hospital, which is responsible for protecting the rights, the safety, and the welfare of the subjects. So the focus of the IRB is subject protection, as simply put as that. So it focuses upon ethics, ensures that things are being done the right way, and that the subject's rights, safety, and welfare are not being compromised in any way. You have local IRBs and you also have central IRBs. We'll go into discussing that later. At this point of time, I just want you to understand what are clinical trials, which are the stakeholders, what are the different phases, and as we go through this, we will then start looking at things in more detail. So now we're identifying different stakeholders. We saw the sponsor, also called the pharma company. Then we saw CROs, which the activities, either partly or wholly, for conducting a clinical trial could be outsourced. Then we had sites, which were the hospitals. Then we have investigators, which are the doctors at the site. They could be the principal investigator or his team, which includes sub-investigators or clinical research coordinators, and of course, things like nursing staff and so on and so forth. Then we have IRBs, or the institutional review boards, or the same thing is also, they are also referred to as institutional ethics committees. In Europe or in India, it's usually referred to as the Institutional Ethics Committee. In the US, it's called the Institutional Review Board. Okay, some more terms that we want to understand. When a clinical trial is conducted, usually you would have someone either from this, whoever is taking ownership for the conduct of the clinical trial, either the CRO or the sponsor, would have a monitor who would go down and monitor the performance of the trial. So this person will visit the site on an ongoing basis and check whether the site is complying with what? What should a site comply with if it's conducting the clinical trial? What do you think a site needs to comply with? Very good, Pooja. The protocol. What is the study protocol? A study protocol is like the master document, which defines the rationale for the study. It says why you're working in this therapeutic indication, why are you working on this molecule in this patient population, why are you conducting this trial, what's your study design, how many subjects are you going to include. It's like a charter for the study, correct? It's saying it defines what are called inclusion, exclusion criteria. You're having very, very strict criteria about which subjects can participate in a trial. Tomorrow, if I wanted to participate in a trial, I can't just walk in and say, hey, please include me in the trial because I also have a similar problem. Remember, we want to minimize confounding factors. And so there are very strict inclusion exclusion criteria for the conduct of a clinical trial. So your protocol defines the objectives of the study, the primary and the secondary objectives. It defines the rationale, it defines inclusion exclusion criteria. It has what's called the schedule of assessments. Schedule of assessments. So your trial would be conducted depending on the phase of the trial. It could run from a phase, from a few months to it could run to a few years. And at different points of time, different procedures, different investigations, etc., will be carried out. So the methodology is defined over there. There are definitions about adverse events, serious adverse events. We'll talk about all of these in a little more detail as we move along. Safety aspects, efficacy assessments, primary, secondary endpoints, and many, many other aspects of the trial get included in the protocol. So it's the most important document for the conduct of the clinical trial. This document is signed by the principal investigator. So going back to my question, what does the site need to comply with? And you said, Pooja, the site needs to comply with the protocol. What else does the site need to comply with? What do we all need to comply with in whatever we do in our lives? Regulations? Laws, regulations, correct? So the FDA regulations, right? Absolutely. And Agatha, you're right as well, GCP. So GCP is good clinical practices. We will speak more about GCP in a few minutes. So the site needs to comply primarily with three things. Of course, the study protocol, FDA regulations, and good clinical practices. I say GCP. So when we're talking about compliance with these, somebody has to go from the sponsor side and ensure that the site is complying with all of these things. Also provide trainings to the site, make sure that the site has received all the important study documents, has received the supplies of the drugs, and so many other aspects that come into this. So that person is called the monitor or the CRA. It's the same thing. And I think, Pooja, you had mentioned you were a CRA at one point of time, right?
Yes.
Correct. So I'm sure you know what a CRA does. So maybe you can share a few things that a CRA does at the site, Pooja? Would you like to share a few things that a CRA would do at the site?
Basically, the CRA, Clinical Research Associate,
Mhm.
goes to the site, and then it will check whether they will check whether all the site is doing everything according to protocol, monitors the study files, um,
Correct. And
monitors
performs other activities like SDV,
yeah, the source data verification. Yes.
SDV, SDR, as well as maybe things like IP accountability,
Yes. IP.
right? Informed consent. So anyway, to make sure everyone's on the same page, the other thing I would like to thank you, Pooja. The other thing I would like everyone to be familiar with, the other term is IP. So what is IP?
Investigational product.
Correct. IP is the investigational product. And what do you mean by IP? When we're talking about investigational product, basically that is referring to the study drug. An investigational product could be a drug, it could be a device, it could be a biologic, it could be any of these. But in the current context, we're really talking about drugs. But all of these are basically your IP. So who will supply the IP to a site? Who supplies the investigational product to the site? The pharma company. And what was the other name? Correct, the sponsor. Get very, very comfortable with the terminology. Pharma company is the sponsor. The site is the hospital. The principal investigator is the primary doctor at the site. And then you have sub-Is and co-Is. The monitor is the clinical research associate. The IP is the investigational product or the study drug. And the other term that we discussed was the CRO, or the contract research organization. And again, we referred to the IRB or the Institutional Review Board at the Institutional Ethics Committee, one and the same. So do you have any questions on this so far? Does anyone have any questions or thoughts?
I had a hard time hearing Pooja say what a CRA does exactly. Could you repeat what she said, please?
Sure. So what the CRA does at the site, remember the first thing, what is the CRA also called? The CRA is also called the study monitor, right? And why is the CRA called a study monitor? Because the CRA monitors. What does the CRA monitor? The CRA monitors compliance. Compliance with what again? Compliance with the protocol, compliance with FDA regulations, compliance with ICH GCP, with clinical practices. And who should be complying? The site. So the CRA or the study monitor goes on the behalf of the CRO or the sponsor to the site and will monitor compliance with all of these three things. While monitoring compliance, the CRA does different things like SDV, source data verification, SDR, source data review. We will discuss what each of these include in a little detail as we move ahead with the session. I'll come to that. And then there's something called IP accountability. IP was also called the, what did we say IP stood for?
Investigational product or
Right.
study drug?
Absolutely. So IP accountability, because every single pill, every single tablet or capsule or whatever your study drug is, whatever form or formulation it is that is supplied to the investigator needs to be accounted for. Remember, this is a research product. It cannot be distributed for commercial use. It cannot be generally passed on to anyone. Every single tablet has to be accounted for. So that is another thing that the CRA does. And then again, the things that the CRA does, reviews informed consent forms. So informed consent, okay, let me come to that in a bit, but the reviews, informed consent forms, reviews, so the investigator has to have an investigator site file where all essential documents are placed over there. So basically the CRA makes sure that all those documents are in place. The CRA trains the investigator and his or her team at, at the start of the study and on an ongoing basis on the study protocol, on GCP and any other things that are required. The CRA also looks at things like IRB submissions, documentation, is everything in place? These are some of the things that the CRA does. Did that help, Sylvia?
Yes, it did. Thank you.
You're welcome. Pooja, did you want to add anything to that?
No, it's good actually. Whatever you said. So basically, there are a lot of stages in when there are a lot of stages like site initiation, then first is site selection, and then site initiation, and then monitoring. So maybe later you'll explain
Yeah,
about.
yeah, absolutely. I'm just trying to give the bigger picture right now.
Yeah, 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 sure.
But absolutely, there are different site visits, and we can go into definitely more detail on all the visits and the correlated activities that the CRA would do. But just to understand some of the important stakeholders, so that's the site monitor, and like I said, through the entire duration of the clinical trial, as per the conventional practice, the site monitor or the CRA, clinical research associate, visits the site on an average once a month to perform all the activities that we have discussed. At the startup stage, as Pooja mentioned rightly, there are different activities involved, but I'm not going to dive into that at this moment. I'm keeping this generic for now. So, going back now to the phases of the clinical trial, we've understood the important players in the clinical trial. We were talking about the phases of the clinical trial. We spoke about phase one, which said this is normally uni-centric or involves a single site. I think now I hope everyone is on one page that we're talking about only one hospital where the trial will be conducted. Usually a small patient population, 20 or 30 subjects. Obviously, you do not want to expose a large patient population or a large number of subjects um, to risk because you still have not established the safety of the drug in humans. So you're keeping your sample size minimal, and because you want to carry out pharmacokinetics, PK studies, when you're carrying out PK studies, basically what you're doing is you're um, taking blood or urine samples at ongoing intervals to monitor the metabolism of the drug, how much is absorbed, how much is metabolized, how much is excreted. So through these blood and urine samples, you get an understanding of how much of the drug is being basically metabolized and excreted from the body. So that's what you're doing in your pharmacokinetic trials. And so normally the subjects would have to be located at the hospital, at the site, for being able to implement this activity, for one to be able to implement this activity. So um, then, then from phase one, you basically move to, and you're, at the end of phase one, you would have typically identified a dose that you consider to be safe in humans. After that, you move to phase two trials. Phase two are also called your exploratory trials. Phase two trials are when, okay, does anyone who has any knowledge on this, would anyone like to share the views on what do phase two trials involve? Um, very, okay, so phase two trials, as I said, yes, of course, at endo, the subject size is larger. So now from 20 or 30, you may have moved to say 200 or 300. You are just one thing to keep in mind through all phases of the clinical trial, you're continuously monitoring safety. That's a given. So phase one prioritizes safety totally. Across the other phases, you do continue to monitor safety. But Pooja, as you rightly mentioned, as you start moving ahead through the other phases, your focus starts shifting towards efficacy. Because you're really happy that your drug is safe. But that's not the only thing you want to establish, right? You do want to establish that your drug is effective as well. So you start moving towards evaluating the efficacy of the drug, and that's where you start going to, or where what you start going to in phase 2. So now you can start increasing your doses, and you start checking that what is the dose that is having, you start trying to optimize your dose, and trying to understand um, you're trying to arrive at the dose which yields maximum efficacy without causing any harm. So at all points in a clinical trial, or when you're dealing with drugs, what you're trying to assess is the benefit risk ratio for any molecule, right? Benefits should always uh, um, outdo the risks. And so phase 2 trials are also often split into phase 2A and phase 2B. So the primary one, the 2A is also called pilot, and 2B is called pivotal. So you're trying to compare, you're trying to identify the right dose, and through 2B, you've kind of established some level of confidence that this is the right dose now that I want to move ahead with. Phase 2 trials involve multiple sites. Like I said, the population is larger, so you would go to say 200 or 300 subjects maybe. There isn't a defined cutoff, but there's a range. It's definitely more than your phase 1. And it could be a longer duration trial. It could be 6 months to a year, maybe. So that's where you're moving ahead with a phase 2 trial. Then you come to a phase 3. Phase 3 trials are also called confirmatory trials. In phase 3 trials, what are you trying to do now? You've moved across from phase 2 to phase 3, from exploratory to confirmatory. You're trying to say, yes, I was fairly confident after phase 2 that this is my dose, which is both safe and effective. And through my phase 3 trials, I'm trying to confirm that. I'm trying to establish it. And so for establishing it with some confidence, I need to increase my sample size so that it has statistical significance and power. And therefore, my sample size in the phase 3 could be something like 2,000 or 3,000 subjects. I may run that across, say, 50 countries, 150 sites or hospitals. And my trial may run for, say, 2 or 3 years. So it's at a much larger scale. The other thing that we need to keep in mind is that we are running what are called controlled clinical trials. So when we are, so what we always do is typically compare our drug either to a placebo or to the standard of care. So a placebo, as I'm sure you're aware, is basically a dummy drug where the drug is not having any active ingredient. The standard of care is basically the alternative therapy that is available in the market for that condition. And we usually start with a hypothesis. What is our hypothesis? You try to establish either non-inferiority, saying that your drug is basically as good as the existing standard of care and you're providing some alternative to that, or you want to establish superiority. So you start with that hypothesis. You would have a lot of statistical, you have something called a SAP, or a statistical analysis plan, where you define your study design, and the plan of your statistical analysis as to how and why you want to conduct that, and how you're trying to establish, either prove or disprove that hypothesis. So that was phase 3. Now, at the end of the phase 3 trial, what you will then submit to the FDA, the Food and Drug Administration, which is the regulatory authority in the US, is an NDA. So what's an NDA? What was an IND again? Correct. Investigational New Drug Application. And Adendo, you're right. and NDA is a new drug application. So what was the difference in what we heard? IND was investigational new drug application. NDA is new drug application. Which word is missing? One word went away. Investigational, absolutely right. So what happened and why is it missing? When we started, remember, when did we submit the IND? At what stage did we submit the IND? Was it before clinical? Correct. So after preclinical and before clinical. Correct? After clinical, preclinical, I'm sorry, and before clinical. So before your first trial in humans, before phase 1. Right? And why were we submitting an IND? We were submitting an IND to obtain permission to conduct clinical trials in humans. That was your IND. Now, as we have completed phase 1, phase 2, phase 3, and we are confident that our drug is both safe and effective, what we want is a permission to. Why is the pharma company conducting all these trials and discovering what's its end objective? Why do you think the pharma company is doing all this? Absolutely right, Sylvia, to get the drug to market. And so you submit your NDA or your new drug application to get approval to market the drug. After you get your NDA approved, you can go ahead and market your drug. So these are two very crucial junctures in your drug development process, moving from IND to NDA. And the reason investigational has gone off from that is because now it is not going to remain investigational. When your NDA is approved, your drug can be marketed because you're not researching it anymore. It's going to be an approved product. So after phase 3, you got your NDA approved, then your drug goes to market. Now it's time to party. We're all very happy that our drug has been approved. However, when your drug is approved, do you think you're good, or do you need to still continue monitoring this drug once it's marketed?
We still should continue monitoring the drug.
Very good. You're all right. Keep monitoring it. And why do you need to continue monitoring the drug? We've done so much of, we have conducted so many studies on that, and we're very happy. We're confident. Remember, we confirmed its safety and efficacy. And why are we monitoring the safety of the drug as yet?
Not for long term, right? Here, long term safety and that was controlled environment, not controlled environment when it goes.
Correct. That's absolutely right. So there are multiple aspects to that. One is to monitor long-term side effects, long-term side effects of the drug. Your trial duration might have been 1 year or 2 years, but you still don't know whether after 5 years there might be some other effects that might be expressed. So you definitely want to keep monitoring that. The second thing is, as you said rightly, in a clinical trial, you have very controlled conditions. So remember, we said we have very strict inclusion-exclusion criteria. We try to minimize all other comorbidities that may exist. You also have a very strict regulation in terms of which medications the subject may be allowed to take. Those that are approved are called concomitant medications, and then there are others that are prohibited medications. So why are we regulating which medications the subject may take? Basically, go ahead.
Drug interactions, potential drug
Absolutely right, Sylvia. So you want to minimize drug-drug interactions because, again, those would add confounding factors to your analysis. But the reality in life is that the moment the drug goes to the market, even though your product label may have a whole lot of information on, saying, what you should and shouldn't take, The reality in life is individuals are taking so many different medications, and you may not have identified certain potential drug-drug interactions. So you want to um, monitor what's happening in the real world. And so there's a lot of support from the FDA for carrying out trial to obtain real world data and real world evidence. So basically monitor what's happening in the real world, look for long-term or hidden or unexpressed side effects. Another aspect to it as well, by the way, is the whole concept of pharmacogenomics and the fact that different populations may have different genetic makeup. And remember, your drugs, so your drugs are basically acting in a certain way by binding to certain proteins, for example, receptors, or they're getting metabolized by enzymes. Now, who's encoding those receptors and those enzymes? Those are genes. So if in certain populations those genes are mutated or those genes are overexpressed, or so many different aspects to that, the way if the enzyme that's involved, say in drug metabolism, in the metabolism of your drug is overexpressed, what's going to happen? If the enzyme that is involved in the metabolism of your drug is overexpressed, is hyperexpressed, what's going to happen?
Your body's going to get rid of the drug fast.
Correct? And so what's going to happen?
It's going to be less effective.
Absolutely right. So you might find out that in a certain population, the drug is not effective at all, while it's effective with others, simply because that population may have variations in terms of the. And what if the enzyme is underexpressed, suppressed, or inactive? What would happen then?
I guess metabolism will be affected.
Correct. In what way? Would it metabolism would be affected, as in will the drug be metabolized at all, or would you see reduced
I
metabolism?
guess it will stay longer in the body, and then if the fellow has problems with the liver or the kidneys, I guess I don't know.
Absolutely right. You brought up very good points, Arindo. So the drug will stay, and right as well, Sylvia, Agatha, the drug will stay for a little longer in the body. And because of that, what's going to happen is it may build up to toxic levels in your body. And that can actually cause significant side effects. And that's why you see that, hey, I'm taking that drug and it has absolutely, it causes me no harm whatsoever, but the person across is taking the same drug, and I had so many complications. These are the things that you want to start understanding and analyzing by monitoring what's happening in the real world. And by the way, that's also what you do try to track to some extent, to the best extent possible, even during the conduct of the clinical trial, where you can have stratification carried out by, it could be by geography, it could be by race. There are different ways of, slicing and dicing the population to understand how the drug acts on different segments of population. But that also brings into what is a very evolving and a happening area today. Sorry for the typo, the area of precision medicine. Uh, okay. So before I now move into the history of human research, are there any questions on what we have discussed so far? Everyone clear on all the phases of the clinical trials, on what INDs are, what NDAs are, what the site needs to comply with? Then what else did we discuss? The phases of the trial. What are the different reasons. We discussed various aspects about the patient population, about different stakeholders, many other things. Any questions or any thoughts or inputs? I see that we have some great experience here on the call. Okay. So if there are no questions, then we can continue with the session now. So let's look at some of the things that happened. If you actually see how all the acts or the regulations in clinical trials evolved, one of the unfortunate things I would say that happened when clinical trials. I'm sorry, Agatha, I'm sorry. Did you have a question regarding the role of coordinators? Was that a comment you were making about something, or is that a question? Review, okay. So you want me to just review. So what does a clinical research coordinator do? The clinical research coordinator is, I would say, the go-to person for the principal investigator. Usually the principal investigator does not directly carry out most of the activities. Usually it would be the CRC who would carry out a lot of the activities. So for example, in the CRC may be involved in the enrollment of the subjects, may be involved in obtaining informed consent. And so let us take a pause for a minute and see what is informed consent. Even before we go there, one of the important things that I would like to ask you all is, can you force anyone to participate in a clinical trial? No, absolutely not. So participation in a clinical trial is voluntary, right? I have another question. So, okay, so you enrolled in a clinical trial, right? At Indu. And suppose you participated in that clinical trial and you enrolled in the trial in January of 2019, and say November 2019, the trial is say a 2-year trial, and in November 19 you said, you know what, I am not having fun, I don't think this drug is helping me, or it's far too much of a headache for me. Can you withdraw from the clinical trial at any point of time? Yes, so these are two very important things to remember, correct? One is participation in a clinical trial is completely voluntary. Nobody can force anyone to participate in a clinical trial. And the second thing is you can withdraw at any point from a clinical trial without any risk to your benefits. You cannot be penalized in any way for withdrawing from a clinical trial. These are two other things that one should remember. A clinical trial involves research. The other important thing is before any individual participates in a clinical trial, you have to perform what is called the informed consent process. Correct? You have to get the informed consent form filled out. So what is informed consent? Informed consent is basically where you are informing the subject about the trial. You're giving the subject information about the study drug, about the nature of the trial, about how many subjects are participating in the trial, about the kinds of investigations that will be carried out, about the potential risks or benefits, what are the alternatives existing, whether the subject will receive any compensation, whom should the subject contact if the subject has more questions, and many other things. But it is absolutely essential that the informed consent form is signed by the subject before any activities related to the clinical trial are performed on the subject. So there is, as Pooja was mentioning, there is something called a screening visit. Well, this is not the CRA's visit, this is the subject's visit. That's the very first time the subject will be screened for participation in the clinical trial. What do you mean by screening? That basically means you're checking whether the subject meets the inclusion and exclusion criteria that are defined in the protocol. So if yes, then the subject could participate in the clinical trial. But for that subject, now what do you mean by something is voluntary? That means you have a choice, right? But what drives that choice? How can you make a choice about something? What do you need to make a choice about something? If I have to choose between A and B, no. My question is, what do you require to make a choice about something? If I have to choose between two things, between A and B, what is the absolutely right, Arindu? You need information. So to be able to make quote unquote an informed decision, you need information. Only then can I choose, because I cannot make a meaningful decision whether I want to participate in a clinical trial or not want to participate in a trial if I don't know what I'm going to gain from it, what risks I'm going to experience, and all the what tests would be carried out on me, what convenience or inconvenience I would experience, whether I would get paid or not, etc. So that's why to be able to have that autonomy, to be able to make a voluntary decision to participate in the trial, you need to be informed. And that's where the concept of informed consent comes in. Is that clear to everyone? Okay, super. So now what I was trying to say is when we're talking about uh, clinical trials or human research, one of the important things that you will see is it's been a very reactive industry, and most of what went wrong resulted in the establishment of new laws and regulations. Let us look at, so when was the Food, Drugs and Cosmetics Act established? The very first regulation that was established for pharmaceutical product manufacturers. That was way back in 1938. Okay. And what triggered the establishment of this FDC Act, or the Food, Drugs and Cosmetics Act? There was the famous sulfanilamide disaster. What are we talking about over here? The sulfanilamide tragedy, what really went wrong? Well, it's also called The Taste of Raspberries, The Taste of Death. It's a very morbid title for this. But in principle, what happened is there was this drug, sulfanilamide, and basically this drug was uh, it was dissolved in diethyl glycol. It was originally used to treat strep infections, streptococcus infections, and there was a scientist. His name was Dr. Harold Watkins, and he thought a good idea would be to dissolve it in diethylene glycol so it would make it easier to swallow, and he added a little bit of raspberry flavor to that to make it tasty and to kind of give it a better flavor instead of having this plain taste of diethylene glycol. And he didn't know that there could be potentially some risk um, that may come in by using diethylene glycol. And so this elixir, the sulfanilamide elixir, was actually provided to patients. Over 600 shipments of this sulfanilamide dissolved in diethylene glycol were shipped. And as you can see over here, more than 100 people died across 15 states. So then it was found out that DEG, or diethyl glycol, was really causing the problem. This is what really gave birth to the Food, Drug and Cosmetics Act of 1938. So 1937, you had the sulfanilamide tragedy. And in 1938 came the Food, Drugs, and Cosmetics Act. And what did this act establish? This act established that manufacturers would have to provide scientific evidence to demonstrate the safety of their drugs before marketing them. Now, this almost makes you cry. I mean, this is the most fundamental thing one would imagine that should have been implemented long ago, right? Why would you market a drug without establishing its safety? But that didn't happen. People lost their lives. And finally, at the cost of more than 100 people's lives, came the FDC Act in 1938, which basically required manufacturers to establish the safety of a drug before marketing it. So that was Harold Watkins, and then we had the Nazi experiments. So these were carried out during the Second World War. We have heard of things like concentration camps and the euthanasia program, or mercy killing as it was called. But basically, those people who were considered unworthy of life, they were basically subjected to the most horrific experiments that you can imagine. What was really, really tragic over here is it was really the most vulnerable who were subjected to all of those experiments. So, who was really, who were the people who were really being subjected to these experiments? They were prisoners, they were physically impaired, they were mentally ill, and all of those kind of, even children and women were being subjected to these experiments. If you even go online and look at some of the videos related to this, you could really, really break down. It's the kind of experiments that were done was simply terrible. And basically, they cause death, they cause trauma, permanent disability. There were experiments like bone transplantation, poison bullets. People were dumped 70 feet inside the ocean just to see whether, at what pressure would they die. Or, I mean, I don't even want to keep on discussing that, but um, these experiments were totally criminal. The other thing is, was there any informed consent taken? No, there was absolutely no consent taken. Do you think that those, when you're talking about prisoners, you're talking about mentally or physically impaired people? These are all vulnerable. Can they demand that you obtain that their consent should be taken? No, they could not. There were different races that were specifically selected, as you know, like Jewish, like Russians and Polish. They were chosen and they were sent to concentration camps. All kinds of vicious experiments were carried out, and so many people died because of that. So these were literally crimes against humanity. And in 1946, there were 23 German physicians against which the trial was carried out. This was called the doctor's trial, and it was called the trial of the USA versus Karl Brandt. Um, this was literally, as I said, crimes against humanity. Now, these trials were basically carried out in Nuremberg, and this led to the establishment of the Nuremberg Code in 1947. So again, you're seeing the reactive nature of what was happening. In 1946, and prior to that, during the Second World War, where all of these experiments were carried out, that triggered a reaction, a response to that, which led to the establishment of the Nuremberg Code. And then, the doctor's trial was carried out, and you have the whole Nuremberg Code, which was established. And there are basically 10 points in this Nuremberg Code that was established in 1947. So, what do you think would be the first and the most important point? I mean, don't bother to read everything that's on the slide. What do you think is the most important thing? Remember, they were prisoners, they were mentally and physically impaired, vulnerable populations. Absolutely right, consent. The first thing was that voluntary participation and informed consent should be obtained for any clinical trial, right? Nobody should be forced into participating in a clinical trial. Then the second part was basically ensure that you're ensuring benefits over risks. So again, the other important thing is you should ensure that you're not carrying out that research unless it is actually going to benefit society and going to benefit the individuals. So you want to ensure that you are not harming anyone. Was there any intent of benefiting those individuals during the Nazi trials? No, right? There was none whatsoever. There was no good intended, no intent of beneficence towards that population. They were purely experiments. They were being treated worse than animals. Again, now this is something so fundamental, but preliminary tests on animals have to be first conducted before carrying out trials on humans. All unnecessary physical and mental suffering should be avoided. No experiments should be carried out if there is a belief that it will cause death or disabling injury. Physicians shouldn't enroll themselves as subjects. The degree of risk should not supersede the benefit. So obviously you want the benefit to be higher than the risk. You should have adequate preparations to protect subjects from injury. So in case, and that is one more thing that a CRA will check when at the initial startup stage, site feasibility assessment, that does the site have the right infrastructure, the right facilities? In case of any adverse events, do you have qualified staff out there to be able to actually treat patients as required? So you should have the proper preparations in place. Experiments should be conducted by scientifically qualified persons. The subject should have the right to leave the experiment at any point of time. As we said, that is voluntary participation without any prejudice, no penalties, no harm. The subject will still retain all rights to his or her regular benefits and to alternative treatment. Cannot decline, the physician or the investigator cannot decline alternative standard of care therapy to a subject if the subject says, I want to stop participating in a clinical trial. And of course, the subject should terminate the study if the scientist, should terminate the study if he or she feels that there is better treatment available. And we'll see some more examples as we move ahead where this actually did not happen. So to quickly recap, the important thing is it's voluntary. You want to obtain informed consent. You want to ensure that you're not unnecessarily conducting this trial if there is existing and better, established alternatives that's better than this. You will definitely test it first in animals. You want to avoid any unnecessary mental or physical suffering. Definitely do not conduct these trials if you know that it could cause permanent injury or death. Benefit risk ratio should be positive towards benefits. Adequate infrastructure and trained staff should be available to treat patients appropriately. Then, of course, the voluntary participation, subjects should be allowed to quit whenever they want to. And in case there is foreseeable risk, then the investigator should stop the trial immediately, or if the investigator finds out that there are better alternatives available. So any questions on the Nuremberg Code so far, or anything that we have discussed? Okay. So if not, I think we'll just take a break. Let's take a break and resume at, say, in 5 minutes. Are we all good with that? Okay, so it's 8:03. Let's resume by 8:08. Hello, can you, yep, can you hear me now? Okay, and you can see my screen, right? Okay, cool. So we looked at the sulfanilamide tragedy, we saw the Nazi trials, and then there is something else that happened. The thalidomide disaster. And this was another major tragedy. It actually happened in 1960. So have any of you heard of thalidomide? Right. And what was thalidomide originally used to treat? What was it used for?
Pregnancy.
Oh, sorry. I know it causes amelia in pregnant women.
Okay, good. Well, it was originally used to treat insomnia. It was at that time considered to be the only non-barbiturate sedative. And it was, because of that, it was a very popular drug. And it was actually marketed in over 46 countries. Okay, and so it was marketed across Europe, Asia, Australia, America, and Africa. So I'm not sure where it wasn't marketed. That was way back in 1957. But what happened over there is that there was an Australian obstetrician. His name was Dr. McBride. And he started noticing that this drug was effective for treating morning sickness in pregnant women, as you rightly pointed out, Adindo. So then he started the off-label use of the drug for morning sickness. And he said, that's great, it's solving the problem. What were the consequences? The consequences that were that it caused significant limb and bowel malformations in the children, in the newborn children, right? And this was called seal limbs, as you can see. It is so tragic. Or also called phocomelia. Is anyone not on mute? Request everyone to stay on mute, please. Okay. So phocomelia, or seal-like limbs, it was basically causing congenital anomalies. And what is even sadder is that almost 40% of the children that were born with these malformations died before they were even 1 year old. So it was a major disaster. And the advantage to the US is there was an FDA inspector. Her name was Frances Kelsey. And Frances basically refused to approve the marketing of the drug in the US because she observed that there wasn't enough data to establish whether the drug could cross the placenta. So basically, this was not approved in the US. In spite of that, actually, this drug did get supplied in the US as well. A fair amount of drug was shipped and sold in the US as well. So all of these disasters then led to yet another amendment. These were called the Kefauver-Harris Amendments of the Food, Drugs, and Cosmetic Act. And this was then established in 1962. You're seeing disaster, regulation, disaster, regulation. That's actually what has happened. Sulfanilamide tragedy. What came after the sulfanilamide tragedy in 1937? What came in 1938? We just discussed it.
Nuremberg.
Nope. The Food, Drugs, and Cosmetics Act in 1938. Then after that, in around 1946, at the time of the Second World War, correct, were the Nazi trials. And now, as you pointed out at Indo, in 1947 came the Nuremberg Code. That's where the doctor's trial was basically carried out. And that's how it got the name Nuremberg Code. Then you have the thalidomide disaster, which happened in 1960. And with that, in 1962 came the Kefauver-Harris Amendments. Now, and you can see some of the malformations that have happened to individuals. It's disastrous. So, what did the Kefauver-Harris Amendments require? They required that you have to establish the efficacy and the safety of the drug before it is marketed. Rocket science, right? I mean, you really needed a disaster like the thalidomide disaster to be able to say that you have to establish safety and efficacy of a drug before marketing it. Seems like semantics. One would have imagined that this should have been there long ago, but it wasn't. And now in this amendment to the Food, Drugs, and Cosmetic Acts came in these clauses. In addition, there was a requirement that drug advertising should disclose accurate information about the side effects of a drug. That should not be, it should not be a false, the false promotion. The facts should actually be disclosed while advertising the product. And finally, in case of generics, basically cheap generics should not be marketed as expensive branded drugs by providing a new trade name. So new brand names and remarketing generic, the whole objective of generics was to reduce the cost of drugs. So the third thing that the Kefauver-Harris amendments basically mandated is, so one was the advertising, one of the first obviously was the establishing the safety and efficacy of the drug before marketing it. And the third one was basically ensuring that generics were not sold at the price of a branded drug by providing it a new brand name. So yeah, so these were the main consequences of the thalidomide disaster. Then after that came the Willowbrook Study. What was the Willowbrook Study? This was carried out in Staten Island in New York in the early 1960s. And the state school at Staten Island was for mentally retarded children. That's the Willowbrook State School. And what was observed that the kids over here were getting a lot of a lot of the hepatitis virus infection. Can everyone hear me clearly? Okay, thank you. So further investigation indicated that what doctors were doing is, now remember, this was a school for mentally retarded or retarded kids. So the doctors mandated that the parents should agree that their kids will be infected, deliberately infected with the live hepatitis virus to actually study the course of the infection in the children. This is unimaginably cruel because it's children here, and they were being intentionally, deliberately infected with the live hepatitis virus. And parents desperately needed to get their kids into the school. There mustn't have been too many options available, so they were actually agreeing to that. Again, this obviously created a lot of disturbance about what was happening, freedom of consent. Uh, should a child's consent also be taken? And the consequences that these were having on the child from a perspective of their developing chronic liver disease and other problems. And the parents were not even told about existing treatment options. So coming to the Declaration of Helsinki. Now, the Declaration of Helsinki was established in 1964. 1962, what happened in 1962? 1960 was the thalidomide disaster. Then what happened in 1962? We just discussed it. Which amendments?
The Kavanaugh-Harris.
The Farber-Harris amendments. Very good. That was 1962. Then came 1964, where basically the World Medical Association established the Declaration of Helsinki. This is literally, as it's been described, a landmark international agreement, a cornerstone document on human research. It basically defined ethical principles regarding human experimentation or human research. This includes the guidelines for the ethical conduct for humans in biomedical research, and it incorporates basically the 10 principles of the Nuremberg Code, and has, I think, more than 7 amendments as of 2013. So several amendments came in place. It also laid the foundations for ICH GCP. We will talk about ICH GCP in a bit, and what basically the Declaration of Helsinki highlighted, for example, the right to self-determination, the right to make informed decisions, and the primary duty of the investigator to protect subjects and especially vulnerable populations. Which were the main things? The right to self-determination, the right to make informed decisions, which outlines the importance of informed consent, and the primary duty of the investigator to protect subjects, and especially vulnerable populations. So the Declaration of Helsinki is a very, very important document, and that was driven by the um, by the World Medical Association. I'm sorry. Okay, so then I want to talk about the Tuskegee syphilis study. So what happened in case of the Tuskegee syphilis study? So where was this trial carried out? And mind you, this is considered to be the longest lasting trial in the world, pretty much. It lasted for 40 years, from 1932 to 1972, 40 years. It was also called the untreated syphilis in the Negro male. And as you can make out from this, it was targeted, the African American population was unfairly targeted, was very wrongly targeted. Almost 600 were African Americans, out of which around 400 who had syphilis and the remaining did not. Basically, they were selected. They were told that they were being treated for bad blood, which was crazy. And then after that, those who actually had the infection were not even told that they had this disease. The others were deliberately infected, and basically, even though penicillin was identified as a cure for syphilis in 1947, these patients or these individuals were neither treated with it, were not even informed that there was a cure existing for this condition. The Tuskegee syphilis, Tuskegee is in Alabama. Tuskegee syphilis study is the longest unethical trial in history. And what was a little shocking is that this trial was sponsored by the US Public Health Service. Not only did so many patients die, but 40 of the wives of these subjects, 19 of the children of these subjects also got syphilis. There was a whistleblower, Peter Buxton, who actually found out this information. In fact, those who actually got to know and tried to object and tried to share the information publicly were deliberately suppressed. Peter Buxton was the whistleblower who really got this information out, and then this became public. In fact, in May, I think 1997, Bill Clinton, the president of the USA at that time, actually publicly apologized for this trial. This then led to the establishment of the Belmont Report in 1979. Again, you're seeing a tragedy and a consequence. 1972 is when the Tuskegee syphilis trial ended. It was a 40-year trial, and this led to the establishment of the Belmont Report. The Belmont Report is also a very, very important document. It was basically created by the National Commission for the Protection of Human Subjects. National Commission for what? National Commission for the Protection of Human Subjects. Excuse me. For biomedical and behavioral research. I repeat, the National Commission for the Protection of Human Subjects for biomedical and behavioral research. So they established the Belmont Report. It was issued in September 1978 and then finally became effective, I think, in April 79. So this was the consequence of it was issued in 1978, September 1978, but it became effective in April 1979. So the year you should remember is really 1979. And there are, you can remember the main principles of the Belmont Report as the acronym RBJ. Respect. R is for respect, B is for beneficence, and J is for justice. So what does respect focus upon? What do you mean by respect? When you're respecting an individual, what are you doing? You're giving that individual respect is you're giving the individual the opportunity to make decisions on his or her own. You're giving that individual autonomy. You're not ordering the person or regulating the person. You're respecting that individual's decision. I think when you're looking at these regulations and ethics, it's really not important about just cramming all this into your head and mugging up dates, but understand what triggered those things. And automatically you will remember the consequences and how those regulations evolved and what they're trying to communicate. So respect, autonomy informed decisions and protection of vulnerable populations, informed consent. This is what respect is talking about. Think of what went wrong in the Tuskegee syphilis trial. Were the subjects informed? No. None of them were told that they were having syphilis, right? So there was no informed consent. Forget informed consent. They were not even told that they were having this condition. Forget, they were not told that they were participating in an experiment. They were not told that there were alternative treatments available. They forget that they were not given them. They were not even informed that there was a cure available for the condition. They were actually told that they were, just hold on a moment, please. Sorry about that. So, yeah, where was I? Yeah. So this was the whole concept about exactly no information whatsoever was shared with them, right, Pooja? So there was absolutely no value being given to those people, right? Because when you respect someone, you will discuss with someone, you'll take someone's consent, you will inform the person. None of this was happening. So that's the first part about respect. And the second part about it is beneficence. What do you mean by beneficence? What is a benefactor? What is beneficence? Forget the clinical trials and Belmont Report. What do you mean by beneficence? Exactly. Kindness or mercy, right? Uh, okay, so want to cause no harm, right? So beneficence has two important things. One is do no harm or cause no harm, correct, Agatha? And what's the second one? Maximize benefits over risks. So you want to basically make sure that in any action that you take, you are in no way subjecting individuals um, to risk. Basically, exactly. So two important parts of beneficence are do no harm and ensure that your benefit versus risk ratio is good. So if you understand this, I think you've understood firstly respect, then you've understood the concept of beneficence, and the third one was justice. And when we're talking about justice, what did we mean by justice? Now go back again to your Tuskegee syphilis trial. What had happened over there? I mean, forget now for a moment about the informed consent and all of those aspects. Which population had been selectively targeted? The African American population had been selectively targeted. Was that right? Correct. Was that right? No, that was criminally wrong, right? That should never have happened. It was this was gross injustice, right? And exactly, of course, this was wrong. And so, what is justice talking about? When you say justice, it talks about the equitable distribution of risk. Exactly, it was very wrong. And what justice says is there should be an equitable distribution of risk. What does that mean? So you want people to benefit, you want to distribute benefits equally, you also must distribute risk equally. It's not that you should target a vulnerable population, that they should face the risk, and based on the findings, someone else should get the benefits, right? What we're really talking about over here in terms of the Belmont report, when we speak about justice, we're talking about the equitable distribution of risk, making sure that everyone has an equal share not only of the benefits but also of the risks. So RBJ again are respect, beneficence, and justice. What we saw in what went wrong with the Tuskegee syphilis trial is there was absolutely no equitable distribution of risk. And with that, I will also like to talk about another thing that is done in clinical trials. Usually, you have a process called randomized. Many times, clinical trials are referred to as RCTs or randomized clinical trials. So, what do you mean by randomization? Again, don't bother about clinical trials for a moment. Just tell me what do you think randomization means? Not selective, exactly, but on by chance or random. So, when subjects participate in a clinical trial, they can be they're randomized. That means you have technologies, for example, IVRS or IRT. IVRS is what is IVRS? Interactive Voice Response System. Or you have your interactive randomization IRT technologies. Basically, what is IVRS? A simple example of IVRS is, you've forgotten or you need a new credit card, or you've forgotten your credit card pin or something of that sort. So you call up a helpline, and they will respond to you. You get an automated voice message saying press 1, press 2 for this, and then you go through that most painful and tedious process, but you finally get you what you want. So they have similar technologies which will result in the randomizations of subjects. So bottom line, what it means is if there are 100 subjects participating in a trial, there is they will be randomly allocated to treatments. So, like I said, usually in a clinical trial, you will have your test drug and you will have a comparator. The comparator or your control could be the standard of care or it could be a placebo. So you're randomizing subjects to either treatment arm, and that's why in that way you're implementing an equitable distribution of risk. Is the concept of randomization clear to everyone? Okay, thank you. So that's one thing. You're also minimizing bias, as one of you rightly pointed out a moment ago. So even if somebody wanted to selectively harm someone or to selectively help someone, that would not be possible because of randomization, right? And the other thing that you also have is called blinding. So what do you mean by blinding? Blinding means basically something that is not visible to you. So the other way of minimizing bias in a clinical trial is through blinding. So you have things called exactly not knowing which treatment is assigned to whom. Not knowing which treatment is assigned to whom. So you have something called single blind, double blind, triple blind. So in single blind, guess who doesn't know? Exactly. In single blind, in case of single blind, the subject does not know which treatment he or she is receiving. In case of double blind, any guesses? Exactly. The subject and the investigator, the principal investigator, do not know um, who is receiving the treatment. And finally, there is triple blind. In triple blind, who won't know? Again, both of these, as well as typically the biostatistician, the person who's going to look at and analyze the data, will not know who's receiving the treatment. What's the objective of blinding? Basically, to minimize bias. Because, for example, if I am the investigator and I know that you're on, say, the study drug, then there is a higher chance that if you experience a side effect, I'm going to conclude that that side effect must have been related to the study drug. And also remember, when a subject, like who hasn't taken aspirin over here? Anyone who's not taken aspirin, everyone's taken aspirin, right? But one also knows and is aware that there were so many cases of counterfeit. And again, please, in triple. Okay, so triple blind is basically when the biostatistician or the person who's analyzing the data is blinded. So before you unblind the data, you first perform the analysis, you draw your conclusions about the efficacy of the drugs. It may be treatment arm A versus treatment arm B, but you don't know whether treatment arm A or treatment arm B is the investigational product. So you perform all the analysis and then you basically conclude that, say, treatment arm A is better than treatment arm B. After you have drawn all of these conclusions, that's when you unblind the data and then you find out whether treatment arm A was actually the study drug or it was the standard of care or the placebo. Is that clear, Sylvia? Okay. So, like I was also just talking for a moment about something like aspirin. And we also know that there have been so many cases of counterfeit aspirin products being marketed, basically just the dummy drug, placebo, being sold as aspirin. And people have consumed that and have actually felt that their headaches went away, that they felt wonderful after taking it. So that's the placebo effect, right? So again, if I am the subject and I know I'm on the placebo, even though I'm on the placebo, I might actually feel better just because I think I'm taking the study drug. So you want to eliminate such a placebo effect. You want to baseline it and you want to make sure that it is actually the study drug which is yielding results. So these were aspects around the bias of a clinical trial. Any questions on randomization, blinding, or the Belmont Report, respect, beneficence, and justice? Okay. Another important thing which I did not end up mentioning also in respect is respecting the privacy of research subjects. And has everyone heard of things like HIPAA? I guess you'd have all have heard of HIPAA, right? And what is HIPAA? What is HIPAA? HIPAA is Health Insurance Portability and Accountability Act. What are you basically trying to do through HIPAA? You are trying to, I mean, you're doing many things through HIPAA, but one of the important things are the regulations regarding data privacy and security. And the penalties today are phenomenal. We have a session on HIPAA as well, on ERES, on HIPAA exactly. There are other regulations called that are implemented by Europe. They are called GDPR. That's the general, correct. I didn't do health Information Privacy and Portability and Accountability Act. And GDPR is General Data Privacy Regulation. If you look at any of these acts and regulations, they are having very, very strict laws today requiring that an individual's confidentiality is maintained. There is something called PHI or Protected Health Information. So you have to ensure that you're protecting the data, because if the information is disclosed, it can impact your employment, it can impact your status in society, and your coverage and so many other things. So bottom line, one needs to protect an individual's privacy and the confidentiality of the data. Exactly, Pooja. You want to ensure that healthcare data is safeguarded. So, okay. So now moving on and to discuss some of the historical aspects of human research and some of the regulations. So this is something very important, CFR, which is the Code of Federal Regulations. So what is the difference between GCP and CFR?
Something which is mandatory.
I'm sorry, could you please repeat that?
Regulations are something which is mandatory, and
Absolutely.
guidelines are something which helps to do, which are only recommended. So
Absolutely right.
Yeah.
Absolutely right. So GCP or good clinical practices, these are best practices, these are guidelines, these are suggestions or recommendations of the best way you should be doing things. But CFR or Code of Federal Regulations is basically the law. It is mandatory. You absolutely have to comply with it. And what applies in terms of what we look at is 21 CFR, which is what applies for the FDA's Code of Federal Regulations, Title 21, is what's applicable to us. And then there are many different parts to that. And we will cover many of those parts like part 50, 54, 56, and many others that we will be discussing, 312, 314, 812, and others. So just hold on a moment. Just hold on a moment, please. Sorry about that. So yeah, so that was CFR. And in 1980, basically the FDA established these regulations. Then you have something called the CIOMS guidelines. CIOMS is the Council for the International Organization of Medical Sciences. Basically, CIOMS published the International Ethical Guidelines for Biomedical Research in Human Subjects. This was established in 1982. When did the Belmont Report come into effect? The Belmont Report came into effect in 1979, right? So now 1980 came CFR. 1982 was CIOMS. 1989 was the Office of Research Integrity. So the Public Health Service, US Public Health Service created the Office of Scientific Integrity and the Office of Scientific Integrity Review. That's OSI and OSIR. And then these were integrated to form the Office of Research Integrity. Correct, Sylvia, 1779. So from there, we have moved on. Next year was CFR. 2 years later was CIOMS. And then further down the line was ORI. Originally, the ORI was formed by the integration of OC and OSIR. What was OC? Office of Scientific Integrity. OSIR was the Office of Scientific Integrity Review. These two merged to form the Office of Research Integrity. That happened in 92. And basically the objective of OSI and OSIR was to address scientific misconduct from funding agencies. Then again, you have the very, very famous common rule. So when you talk about the common rule, you talk about 45 CFR 46. CFR is, of course, Code of Federal Regulations. So Title 45, Part 46. And it has 4 subparts, A, B, C, D. Part A refers to the common rule. And this is what is driven by HHS, Department of Human Health Services. So any studies that are funded by HHS basically have to be compliant with the common rule. This was effective in 1991. So that's for the protection of human subjects. Covers research supported by multiple departments, basically under HHS. So we discussed various things that went wrong, and yet here again, you do see one more disaster that occurred. And here, what we're talking about now is an Asian American student, and this was in 96. This Asian American student this Asian American student was studying at the University of Rochester, and this person basically decided to participate in a study of patients simply to undergo a bronchoscopy for the harvesting of alveolar macrophages. Patient was treated with lidocaine, and there was no regulation in terms of the dose that was administered. The consequences were disastrous again. This led to the death of the subject. 96 is what happened here. And then after that, you have what was established with FDAMA. FDAMA is the Food, Drug, and Administration Modernization Act, or FDAMA, FDAMA. FDAMA established various, it was kind of as the name goes, it modernized the FDA. It put in further regulations to establish the purity, safety, and efficacy of drugs. And before we go to the next example, FDAMA also basically mandated the establishment of clinicaltrials.gov, which is basically the registry for clinical trials that exists today. So all clinical trials, initially that was created as per FDAMA, driven by FDAMA in 97. Then in 2005, you have something called ICMJE. Does anyone know what's ICMJE? ICMJE is basically the International Committee of Medical Journal Editors. International Committee of Medical Journal Editors. They said that if you want to publish any research, then you need to register your trial on clinicaltrials.gov. At the end of the day, both investigators and sponsors want to see their information, their outcomes of the research published. After that, a lot of the industry started registering their clinical trials on clinicaltrials.gov. And finally, there was something called FADA that was established in 2007, a decade later. And FADA basically mandated the registration of drugs, biologics, and devices. So you see 97 FADAMA. FADAMA brought in various other improvements also in the Food, Drugs, and Cosmetics Act. But then came FADA in 2007, which made it mandatory for one to register your trials. As we saw, the trials are basically being registered on clinicaltrials.gov. So how many trials do you think are ongoing today? Have been not are ongoing, but have been registered on this clinicaltrials.gov. What's your random guess?
I had a question. Are the on the clinicaltrials.gov, are they ongoing trials or are they completed trials?
All of them. Some of the trials, so the trial is registered, but some of them may have reached completion and some may be still ongoing, right? So again, what's your guess? How many trials appearing on clinicaltrials.gov? Okay, anything, anyone else? By thousands is random. How many do you think, roughly? As I had checked on the 5th of November, which was yesterday, the number was 321,167. 2 million is a little ambitious, but 321,167. This was covering 50 states and 209 countries. So that's the total number of trials that were registered. And then there is an analysis actually that actually shows you how many are actively recruiting and how many are closed, and gives you a breakup as well if you go and look at the site. So it's interesting that so many trials today are registered on clinicaltrials.gov. It also gives you a split as to how many are US only and how many are across geographies, etc. Then we can talk about another experiment, and we spoke about precision medicine and pharmacogenomics, and there was a trial. This was a gene therapy trial, which was carried out on Jesse Gilsinger. This happened in 1999. This was a young boy. He was just 18. And he was basically being treated for a liver disease. The enzyme was ornithine transcarbamylase, OTC or ornithine transcarbamylase. So in this disease, there is a deficiency in this enzyme. And this enzyme basically is involved in the metabolism of ammonia. So basically when this enzyme is not functioning properly, ammonia metabolism is impacted. So this trial was carried out basically at the University of Pennsylvania at the Institute for Human Gene Therapy at UPenn. And Jesse was injected with a vector, an adenoviral vector, which carried the corrected gene, the gene without the mutation, with the aspiration that this would cure the condition. What happened actually is is he ended up having multiple organ failure, and he died within 4 days. Brain death happened, and he died. So now, what all went wrong over here? Let's see what were the kind of problems associated with this. The first thing was that informed consent was not taken. Well, informed consent was taken, but basically it was found out that this experiment had caused death in monkeys, but this was not mentioned in the informed consent form. So, to that extent, disclosure was not complete. That was the first issue. Excuse me. The trial was conducted by Dr. James Wilson from UPenn, as I said, from the Institute of Human Gene Therapy. What was also observed was both Dr. Wilson and the University of Pennsylvania actually had stakes in the research. They had financial stakes in the research. So that itself is again very, very wrong. So we will study the FDA regulations regarding financial disclosures of FIDS, as it's called, later on. But in principle, you can see that there are multiple things that were being violated. In addition to that, this subject was basically replacing another subject who had dropped out of the trial. So when Jesse Gelsinger was included in the trial, it was found that he had very high ammonia levels. And in that condition, he should have not actually been included in the trial. So you can see multiple violations that happened over here, and this resulted in the death of a young boy. 18 is so young, right? And in fact, if you read the analysis of reports, many people said that the expression gene therapy became a taboo after the Gelsinger trial was conducted. So there was, then after that, the university actually settled, I think, this whole thing out of court, and the family received some major returns based on the same. And you see many more cases. There's the whole Veterans Administration, LA Medical Center, where you see other cases of non-compliance, even for such a respected community. Basically, informed consent was not being taken, research was being carried out, and this was way back in 1999. So again, a lot of the research was stopped, and the LA Medical Center was shut down. Again, very, very respected institute, Duke's Clinical Research Institute, DCRI. Again, there were multiple violations. Things like, especially this was with respect to the IRB. What was the IRB again?
Institutional Review Board.
Absolutely. And what was the function of the main function of an IRB?
To check the safety of the subjects involved in the trial.
Yeah, to ensure the protection of, correct, to ensure the safety or the protection. I like to use this term, RSW, right, safety and welfare of the subjects. Okay, so what happened over here is we will speak about the regulations of IRBs and what all they need to do, but amongst the many things that went wrong is IRBs were not maintaining records of appropriate documentation. So you have to maintain minutes of so I have, before I go to what went wrong here, maybe we should talk for a few minutes about something else. So when a clinical trial is being conducted, right, Do you require the FDA approval to conduct a clinical trial? Right. Yes,
Yeah.
absolutely. Right. Do you also require the IRB approval to conduct a clinical trial at a site? Yes. My next question is, if the FDA approves the conduct of the trial, can you initiate activities at a site without the IRB approval? No, you
No.
cannot. Right. So remember, you're obtaining approval both from the FDA and from the IRB. So what is the IRB basically approving? Which kind of documents do you send to the IRB for approval? Which was the most important document for a clinical trial that we discussed? Exactly, the study protocol. Apart from the study protocol, there is also something called an IB. The IB is correct. The IB is the investigator's brochure. So how is the IB different from a protocol? What does the IB contain? It contains, exactly, it contains all information about the study drug. And who is the end user of the IB? Basically, for whom is the IB prepared? The investigator, right? Remember what's the name? Investigator's brochure. So it's for the investigator. We will be covering in our discussions, we'll be covering, in detail about the protocol and the investigator's brochure. But just to briefly tell you, it contains all preclinical and clinical information, CMC information, um, covers the safety, everything about the study drug. And it also has a section which provides guidance to the investigator about, how to deal with what are the expected side effects of the drug and how should the investigator deal with any potential side effects that are observed. So we spoke about the protocol, we spoke about the IB, and then what's the other document that will be submitted to the IRB? Protocol, IB, ICF, correct. What was the ICF? The informed consent form. So these are some of the essential documents that are submitted to the IRB. Apart from that, you submit a lot of other documents. You could include things like your pharmacy manual and many other documents, but you also submit your advertising material, your compensation. We will discuss that in a separate session. But all of these important documents are submitted to the IRB. The IRB reviews them. The IRB makes recommendations for changes. This process can take even up to 6 months or sometimes more before you finally get all your approvals. So there's a back and forth. I've driven many of these submissions and approvals, and it can cause an acute headache, but it is done in the best interest of the subject. So it's a good thing that happens. And the challenge, though, is that many times IRB meetings are not scheduled at a high frequency. So if the IRB meets, say, every 2 months, and then again, you have to submit documents to the IRB for approval, say a month before. So if they make a lot of changes, then again, the whole cycle goes on and this takes time. But the IRB looks at subject protection. It looks at the language used in the informed consent form. Now, what's so important about the language, right? If you look at your documents like your protocol and your IB, how should the language that you use in your protocol and IB differ from the language used in the informed consent form? Exactly. What you have to use is lay language. What do you mean by lay language? Language that is easily understood by the subject. If I describe complex procedures and use high-fangled terminology in the informed consent form, uh, you can be sure that a subject who is not from this domain is not going to understand all that terminology, right? So the IRB will spend a lot of time not only reviewing the scientific aspects, etc., of the documentation, but definitely how are you conveying this information to the subject? How easy is it for a subject to understand it? And humans who are from the scientific field naturally tend to drift into using scientific jargon. These aspects are also reviewed. And only after you've obtained IRB approval can you proceed for initiating the site. By initiating the site, you mean basically you can start recruiting patients at the site. Okay, so what went wrong with Duke's with the IRB is, so anything that has been approved by the IRB needs to maintain records of what was approved. There's a minimum quorum that needs to be there for approving this documentation. So the quorum was not met. Minutes of meetings were not kept. Documentation regarding approvals was not maintained. There were various other such non-compliance cases that happened, which led to research being stopped. And OPRR, that's the Office for Protection from Research Risks, actually stopped trials over there. And I'm sure that has again got initiated, but this at one time was a serious concern. OHRP, that's the Office of Human Research Protection. Again, OHRP, this was established in 2000. Again, the objective is to protect subjects. Okay, this is the case of Ellen Roche, who was basically a healthy research volunteer who was at Johns Hopkins. She was working over there, actually, I think an assistant or something of that sort at Johns Hopkins. She participated in an asthma research study, and the drug used was hexamethonium. Now, she inhaled that hexamethonium, and then she actually died, I think, within a month. She had complications and she died, and this happened in 2001. Johns Hopkins is again a highly institute. What went wrong over here? Multiple things actually went wrong. So, firstly, the investigator who was involved in the conduct of the trial. What went wrong with this is this investigator basically, he had not performed adequate research. So what do you mean by he had not performed adequate research? Actually, there was published literature which actually showed that the inhalation of hexamethonium can cause significant side effects. He had not bothered to go back and dig deep, and so he was not aware of the potential side effects associated with this drug. Secondly, the informed consent form that was provided mentioned that hexamethonium was a medicine. What should the informed consent form have said instead of calling it a medicine? Exactly. It should have mentioned that it was an investigational product. Right. You're all correct. It should have mentioned that it was an investigational product. So the person, the Ellen Roche, who took that drug would have potentially been misled into believing that this was an approved drug, right? And again, This was an asthma study. So the objective of inhaling hexamethonium was to basically simulate the conditions related to asthma and find out why normal people don't suffer from asthma and what triggers asthma in people who are asthmatic. So that's why she was asked to inhale hexamethonium. The other thing that went wrong is she was an employee at that institute. So potentially, would you say that an employee is a vulnerable subject? No? Yes. Tomorrow, if you're working at an exactly, if there is potential coercion. Tomorrow, if you're working at an institution, at a site, and if your boss tells you, or your manager tells you, hey, we're conducting this research, why don't you participate? There will always be the fear in your mind that if you decline, it could impact your promotion, it could impact your job, it could impact your career progression. So the moment you're, say, an employee or you have students participating in a trial, there is always the perception that there could be potential coercion. So this was one more thing that went wrong. And then I think there were various problems associated with the IRB again as to, minutes being maintained. I think there weren't adequate minutes being maintained, not only for this trial, but when it was investigated and evaluated, it was found that minutes were not being maintained for multiple trials, meetings prior to even this one. So this then resulted in an increase in the number of IRBs, the need to train all IRB members, making sure that they are appropriately trained, that the academic review of protocols. Again, one more thing that went wrong is, yes, the investigator did not go back and research the potential side effects, but neither the IRB bothered to go and check on this drug. So why is it going to the IRB for review? The IRB should have at least reviewed this. The IRB also did not investigate further. So the fact that the IRB should evaluate the safety, the validity, and the design of the study, need to work with OHRP. What was OHRP again? Office of Human Research Protection, and establish a committee to review and revise policies for participation of students and employees. And finally, ensure that all relevant documentation is available and written in a timely and an appropriate manner. So this was the case of Ellen Roche. Also the need to re-review consent forms, collaborate with a pharmacist and a librarian for literature searches, because clearly that was missing in this case. Discuss with the FDA to see if an IND needs to be filed. Update IRBs in case of any changes in protocol, any safety updates. Pharmacists need to be involved in the preparation of study drugs, and you need to have random quality checks to ensure the safety and adherence to best practices. So, one thing that I would like to ask you, does the IRB approve the conduct of a clinical trial at the start of a trial? Yes or no? Yes. Does the IRB have to perform continuing review of a clinical trial, or does the IRB have to perform the review only at the beginning? IRB has to perform continuing review of a clinical trial. So it's not just a one-time approval at the start of the trial. Typically, it has to review the activities of the trial at least annually, if not at a higher frequency. And in case there are any protocol amendments, exactly, they need to be submitted to the IRB for approval. Also, if there are any safety updates, the IRB needs to be informed. And why are we doing all this? Basically, what happens if a protocol is amended? It's revised. There may be some investigations that may be changing, which may exactly decrease or increase, either ways, the safety of the subject. So it's up to the IRB to evaluate and assess whether risks and to the safety of the subject have gone down or have increased. Again, safety updates, as I said, if any safety updates are provided by the sponsor to the investigator, then the investigator needs to share the same with um, the with the IRB. So this is actually a very nice slide, and you can look back. This doesn't go way back to which was the first disaster that we studied, which led to the establishment of the Food and Drugs Cosmetics Act, Food, Drugs and Cosmetics Act. Exactly, the sulfanilamide tragedy, also called the taste of raspberries, the taste of death. So that was way back in, when did that happen? Was it in 1932? Are you sure? Was it in 1932 that the sulfanilamide tragedy happened? Was it in 1937? Because in 1938 was the Food, Drugs and Cosmetic Act. Then at the same time, the Tuskegee syphilis trial had started, right? 1946 was when the whole Nazi trials happened, and then there was the doctor's trial, which led to the Nuremberg Code in 1947. Right? These were all the Nazi war crimes that were ongoing. The Tuskegee syphilis trial had started off over here. And then we go on, we saw the examples of the Jewish chronic disease hospital study, the Willowbrook study with the hepatitis virus being injected into kids at the Staten Willowbrook School. Then what's missing over here? Is something missing? What is missing over here? What? What am I not seeing over here, which we studied, which we discussed, which happened? Did we see? I don't see the thalidomide trial here, do we? Exactly. Right, Sylvia. The Kefauver-Harris amendments and the thalidomide trial. 1960 was thalidomide. 1962 were the Kefauver-Harris amendments. 1964 was the Declaration of Helsinki. Then what happened? Then you had the Tuskegee syphilis trial running all along. And then that triggered 1972, it ended, and 1979 was the Belmont Report. 1979 was the Belmont Report, and then we saw the publication of various FDA regulations that really started off in 1980, and some revisions to these by the Department of Health and Human Services and the FDA. 1982 were your SIOMS guidelines. 91 was the Common Rule, which was the rule published by the Department of Human and Health Services for trials funded by HHS. Here you saw the Hipaa privacy rule. You saw the death of Jesse Gelsinger. That was the first gene therapy trial that resulted in a death. Formation of OHRP, or the Office of Human Research Protection. That was in 2000. So yeah, so we saw these milestones already. We've completed this session. Do you have any questions on this? Okay. I think we will call it a day today. I know it's been a lot of information and a lot of stuff. I hope you have enjoyed this session. Was the pace okay for you guys? Okay, good, great. Well, I hope you enjoyed the session. I certainly enjoyed your interaction, great responses, great participation. And the assessments, I'm not involved in that. You'd probably have to speak with, I say that IRB did not do well. That was why OHRP was found. No, I'm not saying that. I'm not sure where that exactly came from, but OHRP was the Office of Human Research Protection. And assessments and assignments are basically provided, will be provided by Solars. So maybe you could speak to possibly Gayatri around that. And just a quick question before, when we start our next session, would you like to do a quick recap on what we have covered? Because I know we covered a lot of stuff and a lot of content.
Hi, this is Gayatri here. The assignments are in the learning portal under the assignment tab and under the activities tab. So the assignments are there for each module. You can go in, check in, and then finish the assignments and turn in them. Am I clear?
Gayatri. Okay, so the last question I had is, would you like me to kind of do a short recap before we start our next session? Okay, super. Good. Well, I think that's it for today. Hope you enjoyed the session and talk to you soon. Thank you all, and thanks, Gayatri.
Thank you, Dr. Nimita. Thank you all.
Most welcome. Take care. Okay, guys.